Okay, so we're now going to start chapter 14 and 15, where we are talking about genetics. So we're first going to start, this first part of the video is going to focus on chapter 14, where we talk about Mendel and what he learned about genes during his research. And then in chapter 15, which will be the next lecture, we will talk about the chromosomal basis of inheritance. We will do lots and lots of genetics practice problems in class. I'm not going to work through the problems in this lecture, but we will do them in class, so it is imperative that you attend class specifically for these chapters to get the practice doing these genetic problems that you will need for the next exam. Okay, so here are some of your key concepts for the next two chapters. So we are going to talk about the laws of probability. We are going to be talking about the laws of probability and how that governs Mendelian inheritance or genetics. We're going to talk about a lot of the human traits that follow these patterns of inheritance. We will talk about Mendelian inheritance and what that means, and talk about how this relates to the behavior of chromosomes that we know and understand from what we know about mitosis and meiosis. So that's pretty much for chapter 14. Those are the key concepts for chapter 14. For chapter 15, we're talking about these next ideas where we will be talking about sex-linked inheritance. So we'll talk about sex-linked genes and the unique pattern of inheritance that they express. We'll talk about linked genes and how they are inherited together. We will not talk about alterations of chromosome number and genetic disorders, so we just won't have time to cover that in this class. You will talk much more about that when you take genetics sophomore year. One of the things that I really want you to focus on and think about as we go through these next chapters is don't forget about what you learned in meiosis and how those chromosomes behave and how they are passed down from the parent cells to the daughter cells. There are going to be key connections between chapters 13 and chapters 14 and 15 that you have to be able to make. You have to understand how meiosis, the chapters that we talked about in meiosis, relate to these chapters on genetics and inheritance of genes. So that's going to be one of the key things that we're going to be working on in class and thinking through as we go through these lectures. Now, before we get into thinking about how genes are passed down from one cell to the next, there's a lot of vocabulary and a lot of terminology that we have to get through for these chapters. So we're going to start in the next few slides working through the vocabulary. So we have already talked about the gametes, and we know that in the humans, these are the sperms in the males and the egg in the female. So these are our gamete cells. Everything that we talk about in these next few chapters are going to be occurring in these gamete cells, and especially in chapter 15. When we talk about the sex-linked genes in chapter 15, we are talking specifically about the gamete cells. Some of the things that we talk about in chapter 14 will also be true for the somatic cells, but we have to understand what those gametes are and what we mean when we use that word gamete. A couple of other terminology or vocabulary words. These are ones that we have already talked about that you should be familiar with from chapters 12 and 13. So the first one is homologous chromosomes or homologous pairs. We refer to them as homologous pairs in chapter 13. Remember, this is what is separated in meiosis 1. So when we go through that first round of meiosis 1, we separate homologous pairs. So this would represent a homologous pair. So one of these chromosomes is from mom, one of these chromosomes is from your dad. They are two chromosomes that carry the same genes for the same trait at the same location. So one comes from mom, one comes from dad. So this specific homologous pair carries the gene for eye color and the genes for height. And so you can see that both of these pairs have those genes at the same location. Here we have another homologous pair that carries the gene for hair color. Again, both of them have information about that gene on the chromosome at the same location. One of these chromosomes came from mom, the other one came from dad. That's your homologous pair. We've also talked about the terms diploid and haploid. So these are, when we talk about diploid, we talk about chromosomes being present in homologous pairs. So you have two of each type of chromosomes. So for example, in the fruit fly, you have 4 homologous pairs or H chromosomes. In humans, we have 23 homologous pairs or 46 chromosomes. So here we have 2 homologous pairs, and so this is a diploid cell because we have 2 long chromosomes and 2 short chromosomes. And we refer to that number as 2n, so 2 times the number of chromosomes. When we talk about haploid, we have 1 of each type of chromosome present, and we refer to that as n. So here, again, we have our 2 chromosomes, 1 long, 1 short. So again, in your cells, your body cells are going to be those diploid cells. Those are going to be the ones that have 2 of each type of chromosome. Your gametes are going to be your haploid cells. All right, a couple of other terms that we need to know. So when we talk about a gene, when we refer to genes, we are talking about a piece of DNA. So a gene is the DNA. We know that the DNA contains the instructions for making the proteins. We learned that back in chapter 6 when we did the tour of the cell. So when we use that term gene, we are simply talking about a segment or a piece of the entire DNA molecule. If this is our entire DNA double helix, when we talk about a gene, we are talking about one segment of that DNA that contains the instructions for making a specific protein that we want to make. Now, the way that we describe genes is by using the term allele. So I realize this is hard to see for you, so just bear with me here. So what we mean by that term allele is it is the way that we characterize a gene and the way that we describe what that gene looks like. They are typically, we typically use letters of the alphabet to represent alleles, and the key is here for this is this statement. 2 alleles are generally required to describe a trait. So alleles are going to be found at the same location on the homologous pairs. So remember back where we had our homologous pair, we had 1 chromosome from mom and 1 chromosome from dad, and we had the gene for hair color at the same location on those chromosomes. That is where those alleles would be represented. So again, this is a way that we describe a gene, and we use letters of the alphabet to do that. Now, when we talk about alleles, we can talk about dominant alleles and we can talk about recessive alleles. Dominant alleles are usually represented by a capital letter of the alphabet, and recessive alleles are represented by a lowercase letter of the alphabet. Also, when we talk about dominant allele, the definition of a dominant allele means that that allele is going to be expressed in the trait if only one of those alleles is present. So as long as we have one big A, it doesn't matter what the other allele is. That big A is going to be expressed. For recessive alleles, they're only going to be expressed if 2 are present to represent that gene. So the only way we get expression of this lowercase a is if we have 2 lowercase a's, and that way we would express the recessive trait. Okay, so additionally, when we talk about alleles, we can talk about homozygous alleles or heterozygous alleles. So when we talk about homozygous, we also use this term true breeding. So anytime you see that term in a word problem or on an exam, you know that we're talking about homozygous alleles. So homozygous simply means that the alleles are the same. So remember, we have to have 2 alleles to represent each trait, and if both of those alleles are the same, so if we have 2 big A alleles or 2 little a alleles, we say that that gene is homozygous. If our alleles are different, so in this case we have a big A on one chromosome and a little a on the other chromosome, then we say that that gene is heterozygous. So we have to understand the difference between those terms and know what they represent. So you're going to see that in a word problem, a homozygous or heterozygous, and you're going to have to know what that represents in terms of the alleles. All right, also when we talk about alleles, we can talk about autosomal alleles or X-linked alleles. When we talk about autosomals, these are the alleles that are on the non-sex chromosomes. So we'll talk about what the sex chromosomes are in just a second. But these are going to be alleles that are not sex-linked, that are expressed the same and inherited the same in every cell in your body. When we talk about X-linked alleles, so by the way, this is going to be in chapter 15. When we get to chapter 15, we're going to be focusing on these X-linked alleles. These are alleles that are located on that X chromosome. So they occur more frequently in one gender versus another, and we'll talk much more about that when we get to chapter 15. All right, so now that we have an understanding of alleles, we can talk about the definition of a genotype. So when I ask you for a genotype, or you see that term genotype, we are talking about what are the actual alleles, what are the letters of the alphabet that represent that trait. So anytime I'm asking you for a genotype, I'm asking you for letters. So these are all examples of potential genotypes that we can have. When I ask you for a phenotype, and I know that's a little bit hard to see, but basically a phenotype is the observable trait, is what we mean by a phenotype. So what is the trait? Is it tall or short? Is it blue eyes or brown eyes? Is it long hair or short hair? So what does the actual trait look like? So I'm not asking for letters. When I ask for a phenotype, I'm asking for what is the actual observable trait that we see in that individual or in that species. The other thing that we have to pay attention to are the generations. So most of the problems that we're going to be working on, we look at 3 generations. So the P generation stands for the parental generation. When I talk about F1, that is the offspring of the parental generation. And when I talk about F2, that is the offspring of the F1 generation. So for example, if you represent the F2, your parents would represent the F1, and your grandparents would represent the P generation. So if you are down here in F1, you are the offspring of your parents' generation. I'm sorry, if you are down here in the F2, your parents represent the F1 generation, and your grandparents represent the P generation. Okay, so we want to take a little look at Mendel and think about what he did and how he informed a lot of what we know right now. And so Mendel did all of his work on pea plants. So these are examples of some of the traits that Mendel looked at and observed in his pea plants. So he looked at pod shape, pod color. He looked at the shape and the color of the the pods. He looked at the flower color, he looked at flower position, and he looked at length. And so when we talk about these traits in the pea plants, we need to understand the phenotype versus the genotype. So for example, if we were looking at the pod color, we could have yellow pods or we could have green pods. So that would represent the phenotype, what we actually observed. So we actually observed yellow pod colors or green pod colors. When we talk about the genotype, again, we're talking about the allelic makeup of that gene. So how would we represent that gene? So if we had little y, this would be, this would be homozygous because the alleles are the same, and it would be recessive because we have lowercase alleles. If we had uppercase alleles, we would call that homozygous dominant. We're going to look at that in just a second. So here we have some representations of alleles, and we're going to talk about the definitions of these things. So we have 2 chromosomes here, which represent a homologous pair. These are unduplicated chromosomes at this point, but we still have 2 chromosomes. One came from mom, one came from dad. We have a couple of different pairs of alleles on this chromosome. So we have represented here, there's one pair of alleles for a trait. Here's another pair of alleles for another trait. And here we have 3 pairs of alleles for 3 other traits. So we actually represent 5 traits on these 2 chromosomes and the alleles that represent those. For the first trait, we have big B, big B. And so these alleles are homozygous. They're both the same capital letter. And because they are capital letters, we know that they are dominant. So we would use the phrase homozygous dominant to describe an allele that is big B, big B. If we look down here at the F's, this bottom allele, we have little f, we would use the phrase homozygous recessive to represent that one. So again, the alleles are the same, so they are homozygous, and they're lowercase letters, so they are recessive. If we look at this allele in the middle, we have big D, little d. So this is a heterozygous allele because the 2 alleles are different. One is capital, one is lowercase. So we would use the term heterozygous to describe those alleles. Now, Mendel did a lot of work with his pea plants, and we basically, the ideas that he described looking at the inheritance pattern of his pea plants are still very much believed today. They are supported today by what we know about the behavior of chromosomes, and that laid the foundation for what we know about mitosis and meiosis, as well as genetic inheritance today. So mitosis and meiosis were first described in the late 1800s, and then we had to really figure out how that related to chromosomes and allelic genetics and what we know today about the way alleles are passed down from one generation to the next. So Mendel actually stated a chromosome theory of inheritance, and there are a couple of different things that that fall under this theory. So the first thing says that Mendelian genes have specific loci or positions on chromosomes. So every gene that we talk about in the next couple of chapters has a specific location on a specific chromosome, either within the body cell or within the gamete. The second tenet of Mendel's theory says that chromosomes undergo segregation and independent assortment. Now, this is where our meiosis is coming back in. So we know that chromosomes were segregated or separated during meiosis, that we separated homologous pairs during meiosis 1, and we separated sister chromatids during meiosis 2. So we're going to go back and think back about that separation that occurred during meiosis as we think about this chromosomal theory. We also talked a little bit about independent assortment. Now, we called it random arrangement. So independent assortment, when we did meiosis, we called it random arrangement, but it's the same thing. We're talking about the way that those homologous pairs lined up along the equator during metaphase 1 of meiosis 1, and how we get random arrangement of those chromosomes. So the behavior of chromosomes during meiosis actually accounts for Mendel's law of segregation and his law of independent assortment that fall under his theory of inheritance. So we're going to talk more about that as we go through and we do examples in class. So real quickly, Mendel's theory of segregation says that diploid cells have pairs of genes or homologous chromosomes. I think we all can agree with that. We can all support that. We talked about our diploid cells, remember, that had 2 long chromosomes, And I'm just going to draw our two short chromosomes. And so we had the ones from mom and the ones from dad. So this looks a lot like what we did in class back in chapter 13 when we talked about meiosis. So this was a diploid cell because we had two of each type of chromosome. We had one set from mom and one set from dad. We know that during meiosis, those chromosomes, and therefore the genes on those chromosomes, segregate and end up in different gametes. So think back to meiosis. If we take this guy and we send him through meiosis 1, so after meiosis 1, we're going to end up with two haploid cells where we have segregated homologous pairs. So one homologous pair goes to this side, one homologous pair goes to this side, one homologous pair goes to this side, and one homologous pair goes to this side. Now remember, we're not done, so we separate these guys again during meiosis 2. Oops. And again, we now end up with our 4 cells because now we're going to separate sister chromatids. So one sister chromatid goes here, one sister chromatid goes here, one goes here, and one goes here. Same thing for the others. We have one here and one here, and one here and one here. So again, these things are not just the chromosomes, they are the genes that are located on those chromosomes. So now we have not only separated those chromosomes into different gametes, we've separated the genes that are located on those chromosomes and the alleles that represent those genes on those chromosomes to different gametes. All right, now remember back, Mendel's theory of segregation talks about random arrangement. So we have cells that have two alleles for each trait. Again, one comes from mom and one comes from dad. So these are still diploid cells. So again, this is sort of representing what I just laid out for you, where we have our diploid cells here. They undergo interphase. So we duplicate the chromosomes. So from image 1 to image 2, we have gone through interphase, we've gone through S phase, where we duplicated those chromosomes. We go down to image 3. This represents meiosis 1, where we separate those homologous pairs. And then we go down to image 4, where we separate those sister chromatids into the gametes. So the alleles on these chromosomes have been separated out into different gametes. And then we can take it one step forward where we get those gametes joining in fertilization to produce a zygote. So this is all relating back to meiosis that we did back in chapter 13. And now instead of only keeping track of the chromosomes, we are keeping track of the alleles that are on those chromosomes that represent the traits that are expressed on those chromosomes. So we will talk more about this in class, but hopefully this is making sense. Okay, the way that we keep track of genes and alleles when we do genetics problems, and again, we're going to do a lot of these in class, is by using the Punnett square, and we talk a lot about probability. So maybe you remember this from high school when you did genetics problems in high school. But the way we use the Punnett square is we take the gametes from the female and put them on one side of the Punnett square. We take the gametes from the male and put them on the other side of the Punnett square, and then we fill in the square. So for example, in this square, we would have a little a and a little a because we take a little a from the female, we take a little a from the male, and so we fill in little a, little a. Here in this square, we would have big A from the female, little a from the male, and so we end up with big A, little a. Same thing here in this square, we have little a from the female, big A from the male, so we end up with big A, little a. And then in this square, we have big A, because one from the female and one from the male. So you need to get comfortable doing these Punnett squares. We will practice them in class, but you will do them over and over and over again. All right, so I'm going to do one quick problem here, and I'll work this one out here for you in the lecture, and then we will do a lot more of these in class. So this says, in humans, an extra finger or toe is a dominant trait called polydactylism. So again, it's a dominant trait, so we represent it with a capital T. The normal condition is recessive, and so we represent that with a lowercase t. This says we have a normal woman, okay? So if she's normal, we know she has to be recessive. So she has to be little t, little t. If she had a big T, she would represent the polydactyl trait. So she cannot have a big T in her genotype. She has to be little t, little t. So the normal woman carries a child whose father is heterozygous for polydactylism. So the fact that he is heterozygous tells us that his alleles are separate, are different. So not the same, but we have different alleles. And he is polydactyl. So we know he has at least one big T, and we know he's heterozygous, so we know his other allele has to be a little t. This wants to know what is the probability that the child is polydactyl. So we have to set up a Punnett square, and we're going to put the female's alleles on top. So her alleles are little t, little t. We're going to put the male's alleles along the side. So he is a big T, little t. And then we can fill in the square for matching up those alleles. So these are the probabilities of the genotypes for the children of this offspring. So we can look at this. We can see that, oops. Okay, so if we look at this, we see that these top two squares both have a big T and therefore would represent a child that is polydactyl. The bottom two squares are both little t's and therefore would represent a child that is normal. So if we were thinking about probabilities here, we would have 2 out of 4, or 1/2, or a 50% chance that our child would be polydactyl. We would also have a 2 out of 4, or a 1/2, or a 50% chance of having a child who is normal. So we'll do a lot more practice examples like this in class, and hopefully you'll get comfortable doing these Punnett squares yourself. Okay, so Mendel's theory of independent assortment, if we go back to that for 1 second, we talk about the fact that because we have random arrangement or random alignment of the homologous pairs during metaphase 1 of meiosis, we get different combinations of possible gametes. So this is when we are now dealing with a cell that has 4 chromosomes in it, and each chromosome has a different allele represented on that chromosome. So one homologous pair represents the gene that is represented by, or contains the gene that is represented by the A allele, and one homologous pair has the gene that is represented by the B allele. So again, because we can get random arrangement of those chromosomes during metaphase, we get different outcomes in the gametes. So here, in this case, we had a situation where we had, say, mom over here, and dad over here. So I'll put M's and for dad and dad. So our gametes then, we would have mom represented in this gamete, mom represented over here, dad over here, and dad over here. But we know that that's not always necessarily the way that these gametes could arrange. We could have also had a situation like this where we had mom, dad, okay? And so then we get a different possibility of gametes where we have different representations of those chromosomes. So because of the random arrangements that we see during metaphase of meiosis 1, we get different combinations of the alleles in the gametes when they pass down. We call this a dihybrid cross because we are talking about 2 different genes, the gene represented by allele A and the gene represented by allele B. All right, there are a couple of special circumstances when we talk about dominant and recessive traits. So, one example of this is called incomplete dominance. Okay, so I think this might be a little hard for you to see this word. I think it got cut off. But this is called incomplete dominance. This is where neither allele is dominant. And if both alleles are present, the phenotype is somewhere in between the homozygous dominant and the homozygous recessive parent. So a good example of this is in snapdragons. So we have a red snapdragon, which represents a homozygous parent. Let's call this one big R, big R. Then we have a homozygous parent that is represented by the white flowers, and we might call this little r, little r. We cross those two flowers, and we get a snapdragon with pink flowers. And so the genotype for this guy would be big R, little r. So under normal dominant recessive rules, we would think that this would be a red flower because it's got one big R, so we think that that would express that dominant trait, which in our case here is red flowers. But it doesn't. It gives us pink flowers instead. And so this is a case of incomplete dominance where we have something that is in between the phenotype of the parents. So if we were to look at this, if we were to do a Punnett square for our parents, if we had a big R and a big R and a little r and a little r, we would see that every single one of our offspring is going to have this heterozygous phenotype. I'm sorry, this heterozygous genotype. Every single one is going to be heterozygous. And that is true that anytime you cross a homozygous dominant parent to a homozygous recessive parent, you're going to end up with 100% of the offspring being heterozygous in their genotypes. Now, that's not necessarily true for the phenotypes. We'll talk about that later in class. But for the phenotypes, you get 100%, I'm sorry, for the genotypes, you get 100% of them being heterozygous. So all of our offspring would have these pink flowers in this case. We can also think about situations where we have codominance. And this is where both dominant alleles are going to be expressed. So the best example of this is blood typing. So we have types A blood, types B blood, types O, and types AB. AB is the example of where we see this codominance come in. So we can have different alleles that represent each of these different types of blood. So for type A, we can have the heterozygous dominant trait, or the heterozygous dominant genotype, where we have I big A, or we can have the heterozygous genotype, where we have I big A, lowercase I. So lowercase I is our recessive allele here, and I big A is our dominant allele. So because we have both of those, because we have at least the one of the dominant traits, we're going to see the dominant phenotype expressed. Same thing with the B's, except we have the B genotype instead of the A. When only way that we get O, again, is if we have that homozygous recessive genotype where we have little I, little I. However, when we have an allele that is I big A and an allele that is I big B, both of those alleles are going to be expressed. They're both dominant, and one is not going to be more dominant than the other. They're both dominant, they're both going to be represented, and therefore we end up with type AB blood. So the blood typing is an example of sort of an exception to our typical dominant recessive rules, where we get this example, or this case, of codominance.